I will be basically continue with advanced stages of the glycolysis. Okay, now it's clear, voice. Okay, so starting with the glucose, where the first primary reaction occurs, where the first primary reaction occurs is with glucose, where a molecule of ATP is used for the energy. Okay, where a molecule of ATP is used as a energy. So after gaining energy from the ATP molecule, okay, enzyme hexokinase converts glucose into glucose 6-phosphate. Okay, converts glucose into glucose 6-phosphate. Okay, as shown here. Okay, as shown here in the screen. Okay, this is how a glucose 6-phosphate molecule is formed. Okay, moving forward, glucose 6-phosphate will now be converted into fructose 6-phosphate with the help of enzyme isomerase. Okay, to be specific, it is phosphohexose isomerase. Okay, it's a reversible reaction, and the first step which involved hexokinase was basically a regulatory step. Okay, it was basically a regulatory step. Okay, so now moving again, fructose 6-phosphate will now be converted into fructose 1,6-biphosphate with the help of enzyme phosphofructokinase with the help of ATP, okay, which will be converted into ADP, okay, which will be converted into ADP, okay. Again, this is basically an irreversible step. It's an irreversible step which involves ATP for the energy. Okay. Now moving forward again, fructose 1,6-biphosphate, okay, will now be converted into dihydroxy acetone phosphate and glyceraldehyde 3-phosphate with the help of enzyme aldolase. Okay, where a 6-chain carbon of the fructose is broken down into 3-chain carbon molecules. Of one of them is glyceraldehyde 3-phosphate and the another one is the dihydroxy acetone phosphate. Okay, so moving forward, now further, there will be an isomerization reaction between glyceraldehyde 3-phosphate and dihydroxy acetone phosphate. Okay, where glyceraldehyde 3-phosphate will be converted into dihydroxy acetone phosphate will be converted into dihydroxyacetone phosphate, and vice versa, dihydroxyacetone phosphate will be converted into glyceraldehyde 3-phosphate with the help of enzyme thiose phosphate isomerase. Okay, now this reaction decides the fate of the glycolysis. Okay, will it move forward toward the pyruvate molecule or the dihydroxyacetone phosphate will now be further used for other purposes. Okay, it will depend purely on the requirements of the cell. Now moving forward, now payoff phase. The first phase was basically energy was used. Now the second phase, which involves generation of the energy, okay, which involves the generation of the energy. Now the phosphate groups, okay, as we discussed earlier also, are completely ionized at pH 7. Okay, so that each of the 9 intermediates of the glycolysis gains net negative charge. Okay, it gains a net negative charge since the cell membranes are in general impermeable to the charged molecules. The glycolytic intermediates cannot escape from the cell. Okay, none of the glycolytic intermediates can escape from the cell. Only glucose, okay, can enter cell, and pyruvate or lactate can leave the cell. And pyruvate is the end product of the glycolysis. Okay, and how is lactate formed? We will talk in next lecture. Okay, we will discuss it in next lecture. Okay, and why it is not possible for other intermediates to move out of the cell. The reason is simple, because the cell have membranes have a specific transport system that allow only the selected molecules to pass through the membrane. Okay, to pass through the membrane. Now the second function, let's say, okay, as we all know, phosphate is the essential molecule. Sorry, phosphate is the essential molecule for the conservation of the energy. Okay, where they are transferred to the ADP. Okay, now if we add a phosphate molecule to the ADP molecule, it will get converted into ATP. Okay, no doubt the reaction which involves is basically a substrate level phosphorylation reaction. Okay, energy can be generated. One of them is substrate level phosphorylation, another one is the electron transport chain. Okay, in this unit 2 for TY, okay, we are not going to discuss electron transport. Okay, for second year, uh, we are going to discuss ETC in the unit 2 itself. Okay, okay, now moving forward, the phosphate groups also another function can act for the recognition, okay, or the binding required for the proper fit of the glycolytic intermediates. Okay, so this is how they help. Okay, phosphate basically decides the fate of various glycolytic intermediates, okay, by helping them for the recognition and binding of the proper groups. Okay, so now moving forward, now glycerol diethylphosphate will now be further converted into 1,3 biphosphoglycerate. Okay, with the help of enzyme glyceraldehyde 3-phosphate dehydrogenase. Okay, and the reaction which is involved is the oxidation and phosphorylation reaction. Okay, the reaction which is involved is the oxidation and phosphorylation reaction. Now, in this reaction, you can see the structure. Okay, you can see the structure here of the glyceraldehyde 3-phosphate. Okay, now what happens? Now what happens? A phosphate group is added. Okay, phosphate group is added here. Okay, and one more thing, a oxygen molecule is also added. Okay, because it's also an oxidation reaction. Okay, because it's an oxidation reaction also. Now these two phosphate ones will now be added to glyceraldehyde 3-phosphate, but you can see in the diagram itself only one phosphate is added, because it is very simple. As during the process of one glucose molecule, when we start with glycolysis with one glucose molecule, two glyceraldehyde 3-phosphates, okay, two glyceraldehyde 3-phosphates are basically synthesized in the glycolysis process. Okay, so the two phosphate molecules are basically used. Okay, now moving forward, here two NAD molecules will be converted into two NADH. Okay, two NADH. Okay, and each NADH molecule conversion gives rise to a three molecules of ATP. Okay, gives rise to a three molecule of ATP. We will discuss it more during the electron transport chain. Okay, as per new calculation given. Okay, as per new calculation, only 2.5 molecules of ATP are produced per conversion of NAD to NADH. Okay, per conversion of NAD to the NADH. Okay, now moving forward, have you all understood this one? Have you all understood this step? Okay, now 1,3 biphosphoglycerate, okay, will now be converted into 3-phosphoglycerate. Okay, will now be converted into 3-phosphoglycerate, and as I discussed few minutes ago, okay, let's say ATP formation it occurs basically by two ways. One of them is substrate level phosphorylation, and another one is basically electron transport chain. Okay. Now this example given here is an example of substrate level phosphorylation. Okay, it is the first step of the glycolysis where ATP molecules are formed. Okay, where ATP molecules are formed. Okay, where two ADP molecules are converted into two molecules of ATP, into two molecules of the ATP. Okay, where one phosphate molecule from the 1,3-biphosphate glycerate gets cleaved, okay, and joins ADP. Okay, and to be specific, okay, as there two molecules of 1,3-biphosphoglycerate are formed. Okay, so two molecules of 1,3-biphosphoglycerate will be formed. Okay, so two number of ATPs will be formed from two molecules of ADP. The enzyme phosphoglycerate kinase is used for the formation of 3-phosphoglycerate. Okay, 3-phosphoglycerate. Okay, uh, speaking specifically for the ATP formation, almost more than 80% of our ATPs are formed through the electron transport chain. Okay, almost more than 80%. Only the rest are formed through various other processes. One of the main process which involves is the substrate level phosphorylation. Okay, substrate level phosphorylation. Okay, so moving forward, any questions? Okay, now 3-phosphoglycerate will now be converted into 2-phosphoglycerate. Okay, this reaction is basically just a type of isomerization reaction, just a restructuring more specifically. Okay, just a restructuring of 3-phosphoglycerate molecule, which involves the enzyme phosphoglycerate mutase. Okay, which involves the enzyme phosphoglycerate mutase. Okay, where 3-phosphoglycerate will be converted into 2-phosphoglycerate. Okay, and also this reaction is an reversible reaction. Okay, so 2-phosphoglycerate can also be converted into 3-phosphoglycerate. Okay, now 2-phosphoglycerate will now be get converted into phosphoenol pyruvate. Okay, will now be converted into phosphoenol pyruvate with the help of enzyme enolase. Okay, with the help of enzyme enolase. Okay, in this case, a basically dehydration reaction occurs. Okay, because water molecule is released during the conversion of 2-phosphoglycerate into phosphoenol pyruvate. Okay, also this one also is an reversible reaction. One thing more, when phosphoenol pyruvate is converted into 2-phosphoglycerate, there will be basically a rehydration reaction. Okay, there will be a rehydration reaction. Okay, because there will be need of two molecules of hydrogen and one of the oxygen. Okay, so the reverse involves the rehydration one. Okay, now moving forward, phosphoenol pyruvate will now be converted into finally our end product of the glycolysis. Okay, will now be converted into the end product of the glycolysis. Okay, with the help of enzyme pyruvate kinase. With the help of enzyme pyruvate kinase, again this reaction is a substrate level phosphorylation. Okay, second ATP formation reaction in the glycolysis. In the glycolysis. Okay, and this one it's an irreversible reaction. Okay, this one it's an irreversible reaction. Okay, where phosphoenol pyruvate is converted into pyruvate with the help of enzyme pyruvate kinase. With the help of enzyme pyruvate kinase. Okay, so I will just revise the glycolysis. Okay, if you have any questions, you can ask right now. If you don't have questions, please answer no, sir. Now I'm going to start with the fate of pyruvate molecules. I'm going to discuss about the fate of pyruvate molecules. Okay then. Now, as you all know, glycolysis basically occurs in many of the aerobic bacteria. Okay, many of the aerobic bacteria, aerobic living organisms. Okay, but what will happen? Okay, when we discuss about the facultative anaerobes. Okay, what happens when we discuss about facultative anaerobes? Okay, or let's say in an animal cell. Okay, in an animal cell, as all animals you know, all of them are aerobic ones. Okay, all of them are aerobic ones. Okay, let's say due to some other reason, oxygen level falls in an individual. Okay, falls during a in an individual due to various reasons. It may be due to over exercising, or he may have some other genetic problems, or other problems, or during various other functions. Okay, there are variety of reasons that in our body, at some point, at some part of the organ, there are chances anaerobic conditions are created. Okay, so what happens in those cases? What happens when they are facultative anaerobic conditions, and what happens when they are purely aerobic conditions? Okay, so now we'll discuss about the first one. Okay, which is basically a hypoxic or anaerobic conditions. Okay, hypoxic or anaerobic conditions. This particular reaction occurs in facultative anaerobes. Okay, or let's say if due to some reason oxygen level is very low in the system. Okay, this occurs when in the facultative anaerobes. Okay, or due to some reason oxygen level is low in the system. Here the pyruvate molecule will get converted into ethanol and carbon dioxide. Ethanol and carbon dioxide. Okay, and example I have given here is the fermentation of ethanol in yeast. Okay, fermentation of ethanol by yeast. Okay, and this is how bakery products are made. Okay, this is how bakery products are made. Okay, now let's take an example of the fermentation of ethanol as well as carbon dioxide formation by yeast. Okay, you all know for bakery products, okay, you will need a dough, okay, a flour which is being used to develop a dough. Okay, when we add yeast in this dough, okay, a dough rises due to the gas formed in the dough, which is basically produced, which is basically produced by the yeast. Okay, the gas which is formed is basically a carbon dioxide. Okay, is basically a carbon dioxide, and ethanol is also produced, but up to very low extent. Okay, but at very low extent. When we talk about purely of ethanol formation, in that case also carbon dioxide is also produced, but the process can can be controlled. Okay, process can be controlled. Okay, you will study this more. This one in your semester 6 of microbiology. Okay, how can we control ethanol production and carbon dioxide production? Okay, of the yeast. This is how alcoholic beverages are made and bread is made. Okay. Now the second condition, which is basically pure aerobic conditions. Okay, this is basically a pure aerobic conditions. Here two molecules of pyruvate, okay, two molecules of pyruvate are converted into 2-acetyl CoA, okay, are converted into 2-acetyl CoA, okay, where two molecules of carbon dioxide is released. Okay, here two molecules of carbon dioxide is released. Okay, now this one is a classic example of aerobic organisms. Okay, this one is a classic example of uh, aerobic organisms. Okay, because in order to start ECA cycle or citric acid cycle or Krebs cycle, oxygen is the main requirement. Okay, oxygen is the main requirement for their functioning. Okay, so when there are aerobic conditions available, okay, pyruvate molecules will be converted into acetyl CoA, and the final product of the citric acid cycle will be carbon dioxide and water. Okay, carbon dioxide and water. Okay, this occurs in all the living organisms where TCA cycle occurs. And why TCA cycle is important? Why TCA cycle is important? Reason is simple. It is the main source of most of the living organisms to create amino acids, proteins, even the precursor molecules of various other compounds. Also, one of the main energy generating process, one of the main energy generating process of the system. Okay, for the any living organism which is being which utilizes energy. Okay, one of the main energy generating process of the living system. But one thing main, it involves oxygen. Okay, it involves oxygen. Okay, now the third condition, which is basically anaerobic conditions, which is basically anaerobic conditions. Okay, where pyruvate will be converted into two lactate molecules. Okay, this one is basically a type of fermentation again, similar to formation of ethanol. Okay, here the enzyme which will be used is basically a lactate dehydrogenase. Okay, lactate dehydrogenase, which will produce lactate, or one of the common name is also lactic acid. Okay, as you all know, lactic acid. Now, I will give you one of the example in our body also. Let's say Gautam is basically exercises a lot. Okay, but when he exercises regularly, okay, he regularly exercises about 30 minutes. Okay, and one day he decides that today I am going to exercise for about 1.5 hours. Okay, Gautam has decided to exercise for about 1.5 hours, but regularly it is he exercises for about 30 minutes only. Okay, so the Gautam, after exercising for about 1.5 hours, after 2 hours, let's say, okay, he will have pain in his muscles. Okay, he will have pain in his muscles. Okay, why? What is the reason of the pain? The reason is simple. Okay, due to excessive exercising. Okay, due to the excessive exercising, a level of oxygen depletes. Okay, the oxygen goes down in our muscles. Okay, in our muscles, due to which the pyruvate which is formed. Okay, the pyruvate which is formed, instead of going for the TCA cycle, opts for the formation of lactate. Okay, opts for the formation of lactate. Okay, also known as lactic acid. Now, when this lactic acid accumulates in our body, okay, the increase due to the increase in the concentration of lactic acid, okay, due to the increase in the concentration of lactic acid in the muscles, okay, our muscles start paining. Okay, our muscles start paining due to the contraction. Okay, due to the contraction. Okay, this is how pain occurs in our body. Okay, almost all of the students, even myself, okay, all of must have experienced this pain during your exercising sessions, if you are doing, no doubt. Okay, if you are doing these exercising sessions. Okay, if you do not exercise, then this type of pain you will not experience. Okay, any questions? Any questions? Okay, now how the formation of lactate occurs. Okay, and also the formation of ethanol occurs. Okay, this is a detailed reaction. Okay, which is being shown here. Okay, a pyruvate basically is converted into lactate with the help of enzyme lactate dehydrogenase. With the help of enzyme lactate dehydrogenase. Okay, where NADH will be converted into NAD. Okay, where NADH will be converted into NAD. One thing more, this conversion does not involve any involvement of the ATP. Okay, ATPs are not produced. Okay, in this reaction, ATPs are not produced. Okay, now moving forward for the conversion of pyruvate to the ethanol. Okay, here pyruvate will be converted into acetaldehyde where a molecule of carbon dioxide will be released. Okay, in this process, thiamine pyrophosphate and magnesium ion will be used, okay, for the process. Okay, this is an reversible process. Okay, but during the conversion of acetaldehyde to pyruvate, okay, it basically involves carboxylation process, not the decarboxylation process. Okay, now acetaldehyde basically will be converted into ethanol with the help of enzyme alcohol dehydrogenase, with the help of enzyme alcohol dehydrogenase, where NADH will be converted into NADH. Okay, now further this process involves formation of ATP from the ETC. Sorry, I mistake. Sorry, conversion of NADH to NAD, the formation of NADH to the NAD. Okay, sorry, does not involve any ATPs in the process. Okay, any questions? Any questions? Okay. Now, as I had discussed earlier regarding the fate of the glycolysis, okay, in aerobic organisms, pyruvate, I'm just going to read out. Okay, I'm just going to read out. Is it clear now? I'm just going to read out. Okay, I've already explained you fate of pyruvate. Reduce coenzyme oxidase કરવા માટેનું process છે. So, which coenzyme you are talking about? Okay, yes, NADH. Yes. It's one of the process. Yes. Okay. So pyruvate so formed then enters the mitochondria. Okay, where in the case of bacteria there is no mitochondria, only the cytoplasm. Okay, where it is oxidized with the loss of its carboxyl group as carbon dioxide to form acetyl group of acetyl coenzyme A. Okay, and the latter acetyl group is completely oxidized to carbon dioxide and water molecule by the citric acid cycle or Krebs cycle with the help of the intervention of the oxygen molecule. Okay, and this pathway is followed by almost all animals and plant cells, even the bacterial cells. Okay, but most of them are basically aerobic bacteria. Okay, if the supply of the oxygen is insufficient, as in vigorous skeletal muscles, the pyruvate cannot be oxidized further due to the lack of oxygen present in our body. Okay, under such conditions, it is then reduced to the lactate, a process called anaerobic glycolysis. A lactate is also produced from the glycolysis in anaerobic microorganisms that carry out lactic acid fermentations. Okay, as I have explained this in the earlier session. Okay, now in some of the microorganisms, which is basically a case of facultative anaerobic organisms, okay, the pyruvate formed from the glucose is transformed anaerobically into ethanol and carbon dioxide called a process called alcoholic fermentation. Since the living organisms first arose in an atmosphere devoid of oxygen, anaerobic breakdown of glucose is one of the most ancient type of biological mechanism for obtaining energy from the organic fuel molecules. Okay, from the organic fuel molecules. Okay. Now, It's the time. I think I should end lecture right now. Okay. Please join me in the next lecture. Only 2 minutes are remaining in this lecture. Okay. Everyone, please join the next lecture. Comment your name for the attendance and join the next lecture immediately. Okay. Comment your name for the attendance and immediately join the next lecture.